Okay, welcome everybody. I think we are right on time, and I see that many people already joined. So we will start because we have very interesting webinar coming up. So good afternoon and welcome everybody. Today we will be talking about new solutions for mechanobiology studies. And we have 4 experts in the field that will be sharing with us the latest advancements on their research and also their technologies. But before we go with our speakers, I wanted to explain that on the right side of your screen, you will see a panel where you can go to the chat option and you can type all your questions or the comments that you may have during the webinar. We want to keep this like in a very interactive. So some of the questions will be answered on the chat. Some other questions I will be reading them and direct them directly to our speakers so they can reply to you. And just please don't be shy, and you can type it using the chat all the time. And also, I want to thank Massimo Basali because this webinar, he helped us a lot with the organization, and it wouldn't be possible without him. He is also an organizer of the Nanoengineering for Mechanobiology Congress, and he's also working at the James Watt School of Engineering at the University of Glasgow in the UK. And we are very happy to have you here, Massimo. So I would like to give you the word, and you can also welcome everybody.
Hi, Noelia. Thank you very much for your kind introduction. Sorry for being slightly late. So I had to understand how to switch my camera on, which is normal. And so 1 year of lockdown was not enough to understand how to use all this technology. Sorry. But so yes, I'm happy to open, not taking much time to present anything, just to recall everyone that if you are curious to know about technology and how technology can be adapted to study mechanobiology, we organize this little conference every year, which is called Nanoengineering for Mechanobiology. And next year will be at the end of March 2022. We hope to be back in presence and organize a beautiful meeting all together. If you are interested, you can go to this website, which is here. So the other way around. So, but it's n4m.mechanobiology.eu. You can find a lot of information there. And thank you for joining this event. I'm really intrigued by the talk and the mix of imaging and mechanical manipulation of samples as well. Thank you.
Yes, thank you, Massimo, for your words. And I think it's very interesting to also join this conference N4M. And now we can move to our first speaker. So because we have 4 speakers today, I think I can say to Imran that now you can also request to join the room. And I will give the introduction for Dexter Manalili, that is our first speaker today. And he's a PhD candidate at the VU University of Amsterdam, as also at the University of San Carlos. He has a master's degree in physics, and during his PhD, he has been developing a setup that is combining the real-scan confocal microscope with the indentation technique, and he's using this setup to measure physical properties on cells under compression. And I think that today we'll be sharing very interesting results with us. So welcome, Dexter, and thanks for joining us today. You can share your screen now.
Okay, hello. Okay, let me try. Is it working now?
Yes, it's
okay.
working.
Okay, good afternoon, everyone. I'm Dexter. I'm equally nervous, but excited to present to you what I've been working on for more than a year now. So it's a new mechanical measurement technique, which is called a confocal microscopy-assisted ferrule tap indentation technique. So there are 4 mechanical measures of materials. They are listed below, but actually in this list, you only need 4 of them. And we are going to focus the two below, the last two, can be derived from the other measurements. So we'll focus on these four in this research. So we'll start, before we go to the compression of soft biomaterials, we're going to review the compression test for regular materials, like engineering materials. So you have a force, down force, to compress until the material or the sample compresses. And you get this graph where the axes are stress and strain. And from the stress is computed as a force over the area, while the strain is just the elongation of the sample in axial or in lateral. Axial means that it's in the same direction as the force that you exert. So from the slope of this, from the stress strain graph, you can get from the linear part, from the linear elastic part, you can get the Young's modulus. And from the ratio of the strains of the axial and the lateral, you can also get the Poisson's ratio. One thing to note is that for compression tests for these materials, for engineering tests, they use at any point in the compression, the area that you use for the stress is actually the same initial area. So in a way, this is actually not the true stress, which later on, in this experiment, I will provide, or we will provide the true stress and also the true strain. However, there are challenges when it comes to dealing with soft biomaterials. One is that they are too different from one another. So there are different components and within within the material with different sizes and different shapes. But each of them have important local properties that are factor in the global scale of the measurement of the entire sample. Another one is that they're too soft. Just the weight of them can can deform the entire sample. They are responsive to minute forces, and therefore they demand higher sensitivity when it comes to measurement. And of course, they are too small. So they are the deformations. So deformation is directly proportional to the initial dimension. So if your initial dimension is too small, then you would expect that the deformations are also really small. So therefore, you would also need require a precise measurement. And the conventional optical techniques that we use to measure may not be applicable anymore. So again, these are the limitations when testing soft biomaterials. So inhomogeneous, they are in the microscale, and they are very compliant. And currently, there's no universal method to measure soft biomaterials. We do have range of values for the different properties that we've shown before. And yes, so this looks like a problem, but I think for the community, this serves as a challenge, because this is an opportunity for creativity and innovation. So we propose a solution. So the group here, the group of Davide, has developed this indentation technique, where you have a cantilever and a probe on top, and the cantilever bending is detected by the optical fiber, as you can see in figure section F of the figure. That is an optical fiber directly located in front of the gold-coated cantilever. So small deformations of the cantilever, or small formations, can be detected using an interferometer. Later on, I will show you the entire setup. So this is the indenter that we're going to use, a ferrule top indenter. So this is developed by these amazing people. They are the previous researchers working on this, and they have multiple publications on this one. So why use this? This is an all-optical millimeter-sized monolithic device. So they're good for hydrated samples, also good for in vivo, in vitro experiments. And it can detect up to forces down to nanonewton. And you can even use it. Say, for example, in this part, they're using a spherical probe, which you can use for localized measurements of mechanical properties. And we are going to combine this indentation technique with the confocal microscopy. However, there is one thing that I have modified in this setup. So instead, in the section F, instead of using a spherical probe, I'm actually going to use a flat probe, and that will act as a parallel plate compression test setup. So why use confocal microscope? Because you can use the optical sectioning capability to simultaneously measure different structures, and you can have a resolution of up to around 200 nanometers, depending on your setup. And in this experiment, we are actually using the RCM by Confocal NL. So this is the entire setup. The most important part is highlighted in red. So that is the entire indentation setup. As you can see here, there's a gold coated cantilever, which acts as a reflecting surface for the laser that goes through the optical fiber, and that optical fiber goes to the interferometer, which then goes to the lab view. So all the mechanical parts of this experiment is controlled by the LabVIEW program, which is developed by the group as well. And the optical part, the microscopy part, is developed by controlled by the RCM box and together with the micromanager. So this is a very important, the optical fiber shooting the laser onto the gold-coated cantilever, because this makes sure that the movement of the cantilever as it indents or as it pokes or touches the sample is properly measured. So you can also use, so this is the actual setup. So this this setup has three indentation profiles originally. So we have load control, wherein we can set for a certain period of time that the loading of the piezo is you can set it to a certain value and it can remain in that value for a certain period of time. The second profile is the cantilever controlled, wherein the whole indentation system tries to make or keep the cantilever position constant for a user-defined time. And lastly, which is the most important part of this profile, is the depth-controlled profile, which makes sure that the deformation that you do on the sample is kept constant. So that means that your cantilever and your piezo are constantly adjusting themselves so that they can control or they can keep that level of indentation constant at a certain user-defined duration. So that will play a significant role later in my experiment. So this is the compression profile. So this is where the depth-controlled profile of indentation comes in, or in my case, compression profile. So I set compression levels of compressions, and after it goes to that certain level, it stays on that level of depth, and then so that just enough time that I can take images of the entire sample, and it goes through until a certain limit. In our case, we deformed our sample up to 36 micron. So this is when you compress, and then you have like a step. But in our case, it's like small steps, and then stay at that level. We take images, and then we analyze them. So the first sample that we had was actually a BV2 cells. So we use host 33342 to stain the DNA inside the nuclei. But they were too small for our target strain, and we cannot actually detect, or we cannot, at least for our setup, the effect the resulting deformations were not enough for us. They can provide us data, but not enough data points that we can be sure about the results. So therefore, we concluded that okay, we needed a bigger sample, and that's when we found the fixed bovine oocyte, which is around 150 to 180 micron in diameters, and measured 60 to 80 micron in height, and that is already on the bottom of the substrate. So we fixed them, permeabilized, and then stained with Invitrogen cell mask 488. So this is the bovine oocyte from top to bottom at 5 micron interval. However, when we took the confocal images of when we scanned at the Z-axis, top to bottom of the oocyte, the fluorescence outside the cytoplasm is actually less than 100 times than that of the inner part, the cytoplasm, and it almost blends into the background. So what you're seeing right now is just the cytoplasm of the oocyte. So we measure them. So measuring the width, the heights, and the area, we use Fiji, and that is after we segmented them. We processed and threshold the images, the confocal images, the scan. So the red part actually is my attempt to to obtain the profile of the zona pellucida, which is outside the cytoplasm. But as you can see, there is some curve that in the attempt, but you can also see that the other parts as well is included in the thresholding. So it's not a reliable method for getting the zona pellucida. But if you have, if there's a way that you can fluoresce the zona pellucida much higher, then that would certainly certainly help in measuring as well the zona pellucida part or the entire cell itself. But for now, we're going to measure instead just the cytoplasm, which is inside.
Dexter, sorry that I interrupt you. Can you please, one of the attendees says, can you please hide the icon that says Webinar Jam is sharing?
Oh, sorry.
Yeah,
I'm sorry.
Thanks. Okay.
Thank you. So all the physical dimensions are measured by a Fiji, and the load, the piezo cantilever movements are measured via the LabVIEW program, and also which is connected to the interferometer that is constantly detecting the movement of the cantilever. So these are the deformations at different levels. So the first quadrant, one is from the unindented or uncompressed oocyte. And then you can see the progress as you indent deeper. Then you can also see that how it changes. So how do we determine the Poisson's ratio? Well, we already have the strain, the deformations. So we already have the axial, and from the measurements of the widths and the height, you can already get the axial and the lateral deformation. So if you plot them out, then the Poisson's ratio is actually directly measured from the graph. So you just fit, and then from the 15 measurements that we had the result of the Poisson's ratio of the cytoplasm is 0.22. And determining the Young's modulus, again, is directly measured, directly calculated from the slope, because one, you get the force from the loading of the piezo, which is noted by the LabVIEW program. The area is measured again from here, from the one in the center. We measure the widest area of the entire scan, because we measure it from the widest area because that's also where the highest deformation you can get, and it is easier to measure from at any depth. So we got the Young's modulus, an average of 261 Pascal, which is within the range of the existing how it goes, existing measurements. However, measurements for the cytoplasm and not the entire oocyte is rarely found. So this is something that we can check as well with other techniques. So in conclusion, we have introduced a new technique, a new method of measuring mechanical properties of soft materials, or soft biomaterials in this case. It is compatible with the microscale biological samples, such as the cell, the oocyte that we have shown. It can be used to measure different structures simultaneously. That is, if you have an optical sectioning capability that is given by the confocal microscope, or you can also use a 2-photon excitation microscopy. So you can get different dimensions from different locations of different structures, and then you can measure them directly. And in measuring the Young's modulus and the Poisson's ratio, I think this is important that we measure them directly in this most simple way that is free from modeling and fitting parameters. And it is also considered, we consider this to be a supplement to other micromechanical techniques such as indentation and other dynamic tests, because we can use this first, for example, to measure the Poisson's ratio or the Young's modulus before going into dynamic test, which is, if you remember from the start of this talk, is the first purpose of developing the indentation technique. So we can surely just replace the probe with the spherical one, and then you can go to measuring localized. So in a way, this study is linking the global, in a way, global measurement and localized measurement from indentation. And if you have more questions after this, you can go to the room afterwards, where you can ask me questions about this research. Thank you for listening.
Thank you, Dexter. It was a nice presentation, and I see some questions coming, so maybe we have time for one. And as you said, we have later another room prepared for discussions. So this question is from Matis, and they is saying, how does the confocal work beyond the penetration depth? Because for him, it's like the part of the oocyte doesn't look fully resolved.
Yes. About the resolution, this we are using, how do you call this, 10 times. So it has a longer field of depth of field. So you can, yes, that
working
definitely,
distance,
the working distance as well. So you can, it has a balance of resolution and also how it can go through the sample. That is one thing that is for sure needs to be checked as well, the balance between that one. So it's not fully resolved, but as I said, it is measurement of the entire cytoplasm, not the smaller parts in there. But if you can always use higher numerical aperture objectives to have more resolution. But it has a cost. It costs you the field, the amount of thickness that you can scan in your sample. So for us, we used 10x because it is a relatively thick sample, and in this proof of concept, we didn't mind the resolution because we want to have just the entire cytoplasm.
Okay. Fortunately, we don't have time for more questions right now, but I do see more in the chat, so we will address them later or in the room that we have prepared for the discussion. So now we can go, thank you, Dexter, and we can go to our next speaker. That is Dr. Imran. You can start sharing your screen maybe while I introduce you.
Yeah.
And she is an assistant professor at the Department of Physics and Astronomy at the University, the VU University Amsterdam. She obtained a PhD in electrical and electronics engineer at the University of Twente, and her research now is focused on the photonic integrated circuits and cell mechanics. So we are ready to listen to your talk. Thank you, Imran, for being here.
Thank you. Thanks a lot. Can you see my screen properly?
Yes.
Okay, perfect. Thank you. Thanks a lot for the introduction. Yeah, I would like to talk about some new technologies that we are developing in my group to measure the cell mechanics. But to just first start off, I want to show you just showing you a picture, an artistic picture. If it moves. Yeah, an artistic image of a cell, sorry, to let you understand the importance of the mechanics of a cell, why we are interested in that for those who are not already in the field. So this looks like a very complicated machinery indeed. It's a very nice picture. I really like it. And the mechanical properties of this machinery have really fundamental importance, not only in normal conditions, but also on pathological conditions. And over the years, what we have learned is, let's say, if you take the pathological condition like cancer, a cancer cell is way more softer than a normal cell. So we have learned that. And on the other hand, what we have also learned is during fertilization, an egg cell, an oocyte, allows only one sperm cell to penetrate through, and it blocks the others by hardening its outer membrane. So these findings are quite exciting and very interesting. That's why I started to steer my research direction towards cell mechanics. So in the past, but in general, my motivation is basically finding solutions to real life problems. And that is why I'm interested in two applications, taking the two extremes, as I just told you, with cancer, the pathological condition, and the normal condition, which I will talk in 30 seconds maybe. So the first thing, the cancer that we all know is becoming a pandemic. So if you look at the numbers, the statistics that I've written down there, it's going to hit us one way or another. So the crucial thing here is to find the right technique, right treatment technique. So immunotherapy is considered as one of the future of the cancer treatment. And there's even a very, very surprising concept, which I realized that recently, is that trigocytosis, which I'm very interested to just look deeper into it. It's basically called the nibbling of cancer cells, which means there are neutrophils, which are the white blood cells, are biting off the cancer cells until they die. But this is nice, super nice, but what we still don't know is why the neutrophils can bite only certain types of cancer cells. And since this is a mechanical rupture of the membrane, I'm interested to see the relation between the mechanical properties of the cancer cells and why this trigocytosis process is not always 100% effective. So this is one application that I'm very much interested on the pathological side. And on the other side, there's yet another problem which we are not so much aware of as like the cancer case, which is infertility, but it's a big problem as well. Like we have 186 million people, million couples or infertile couples all around the world. The nice thing is there is a treatment, in vitro fertilization, but still, it's a very frustrating treatment technique. I know it personally, and I also know that the success rate is quite low. So one of the reasons why that is low is basically there's no quantitative way of measuring the quality of the embryos or the oocytes to start with, the egg cells. So, but if you look at the literature, what you see that is the properties of the mechanical properties of the oocytes and so the embryos can be used as a biomarker to indicate their viability or their competency. So knowing this fact, I would like to also investigate. It's one of my research application in my research to look at the IVF treatment itself, starting from the oocyte and its mechanical properties of an oocyte at different stages of its development, like maturation, fertilization, and forming an embryo. So these two things are the applications that I have in my mind in my research group. So far, we have already started with IVF. Dexter is also working with me. As you also saw that we have some oocyte experiments that I will also show you some oocyte experiments in my next slides. The cancer side is still in development. We are still developing the idea because the cancer cells are quite smaller with respect to an oocyte. An oocyte is 100 to 140 micron size, easier to handle, even though the scale is micron. Luckily, I have some previous experience working in the field of mechanics coming from my postdoctoral research time. I had the privilege to work with a very nice, unique tool called OCT vibrography, optical coherence tomography vibrography system. And in a nutshell, without going into details, it's basically the combination of interferometry with acoustomechanics. And with that technique, you can observe the very nice standing wave patterns on the surface of the sample that you're looking at, which was a bovine eye glove in that time. But you can also see that the sample is quite big. And if you try to miniaturize it, you need to put quite a bit of effort, and specifically speaking, this. So after my return to Netherlands, after my postdoc, I started to developing new techniques aiming for single cell measurements. So one of the techniques, which I'm not going to talk today, but it's basically it's very much relevant related to this idea of combination of interferometry with acoustomechanics, but in a micron scale level. So we have the preliminary data, but for time constraints, I will just focus on yet another very nice technique that we are developing together with Optics 11. So they are the main driving force behind this idea. And it's basically, we call it optical interferometry-based micropipette aspiration. And in my research line, I always try to combine light somehow with whatever I do, because I believe the strength of light, it's super powerful. And in particular, interferometry is it's always fascinating me how accurate you can measure things with that. So in this project, this optical interferometry-based micropipette aspiration project, I don't know how much you know about the traditional technique, but it's basically you have a cell, and then you have a sample, and then you have a micropipette, and you start applying negative pressure, and then you get some part of the sample inside your micropipette. And by using a camera on top, you analyze the images, and then from there you deduct the aspirated length. If the sample is stiff, then you see a little bit of aspirated length, and if the cell is soft, then you see quite a bit of part of the sample is aspirated towards the inner part of the pipette. So depending on this length, you can estimate the stiffness values of the samples. So this is a limitation, quite a bit of limitations, to be honest, because now you need to aspirate it quite a bit so that your camera could be able to capture this aspirated length. That's the major drawback. But we solved this problem by combining it again, the superpower of light, interferometry, and signal processing attached to it as well, and some modeling, simple modeling. And we turned it into a product, a fiber optic sensing technique that can give you sub-nanometer resolution. So the resolution-wise, spatial resolution, this is what we're talking about, is completely different than what you can see from a traditional micropipette aspiration. Now, with this high sensitivity, high spatial resolution, you can see even the membrane deformations. You don't need to aspirate it too much. And the most critical thing here is we are planning to work with very delicate samples like oocytes, embryos. This is super important that you're not damaging the sample. And how we achieve this is, we can also call this whole approach as like a all optical readout technique, and it's basically combining a very well-known technique, which is Fourier domain optical coherence. I would say tomography, but we're not doing imaging, so we could say optical coherence reflectometry. And we also borrow some post-processing techniques from there, and by combining this already developed nice techniques, we managed to get this nanometer scale vibrations. So the core here is basically we placed an optical fiber inside the micropipette, and also inside the pressure sensor. This is the membrane which is measuring the pressure, and this is our sample. And by using the end facet of the fiber as our reference point, and also the interfaces at each interface of the sample, we create cavities. Here the cavity is between the fiber facet and the pressure sensor membrane. So these cavities are well known. because these are optically defined. And if you start sucking the sample inside your micropipette, you can change this cavity length, and you can easily follow it using some post-processing techniques. So this is basically what we're doing here. But as I said, interferometry is super powerful. So it can give you nanometer scale vibration changes, displacements. So what we do there is, okay, we have several cavities, not just one cavity, but taking the Fourier transform helps us a lot to decouple all these cavities from each other. This first cavity is basically the pressure sensor, it's the strongest one. And then we have the cavity, which is between the fiber facet and the first interface, the membrane interface of the sample we're looking at. And we also have some other contributions coming from the internal reflections, okay? So there's a formula, it's quite well known, and first what we do is we know the pressure sensor, we can look at it, we can extract its phase, we can also look at this peak, we can extract its phase, and there is a relation between the displacement and the phase. And if you look at this with a very simple formula, if you know the phase of your measurement, you can also calculate your displacement, and by doing it so, you can very precisely know what your displacement is, even though you apply very small pressure levels, okay? Because we can detect it. The cavity size is changing, and we can really detect it. Here, the core is the probe design. So the probe is a crucial part. By probe, I mean the probe combines the pressure sensor and also the displacement sensor. It starts with a ferrule, and it's combined with a mating sleeve. And then we attach the pooled glass capillary. This is all custom made, by the way. And there's a 3D printing house, printed housing to keep everything it's more stable altogether, let's say. And the core thing, yeah, sorry. Also, we have an inlet outlet for the water reservoir, so the water comes in and goes out. And on top of that, the core thing here is the fibers, optical fibers that we place for the pressure sensors part, and also for the displacement sensor part. Okay, so this is the pressure sensor, how it looks like. There's the fiber inside, and also since the fiber and the tip of the micropipette, there's a distance, as you can see. You cannot get all the way to the end, otherwise you will block the flow of the water, which is necessary for capturing the cell. There has to be a little bit of a gap. But that's also affecting the beam spot size. That's the focus spot is always preferential in these experiments to increase the sensitivity. That's why we placed the green lens before that, and then afterwards we combined it with a single mode fiber, and we made sure that we are looking more or less with the focused beam, or at least a depth of focus which is not really dramatically decreasing. So we made this also custom made. Around somewhere here we have a green lens, and yeah, also this is what this is also what you get in reality. It looks like a commercial thing that you can buy, but it's not. It's made by an excellent PhD student. So, and nicely, this work we published it very recently. That's why we're very proud of it indeed. So the work has been published together with Hilda Ardema. She's from Utrecht University Veterinary Medicine Department, and we have a very strong team coming from the Optics 11 as well. And Massimiliano is the core person in the whole work. So I also need to show you some results before advertising. It's so much a thing. We have worked with several samples, and we keep trying to increase our portfolio. And if you have any suggestions or any idea, you're always welcome. We work with oocytes, organoids, microgels, fish eggs that I didn't put here, alginate beads, yeah, some polymer stuff that we received from our partners from Twente. And among them, as I said, IVF is our core thing. So we look at the oocytes at we try to look at oocytes at different stages, mature, immature versions, let's say. As I told you in the beginning, this oocyte, the mechanical properties of the oocyte, mainly the membrane, outer membrane of the oocyte, which is called zona pellucida, can be used as a biomarker for finding its competency or the level of maturity of it. because as it gets mature, the zona pellicida gets softened, so that a sperm cell can penetrate easily. Okay, so this is a known fact, and we try to see this. So far, our experimental setup is not ready yet for these things, but we're working on it now. So again, we get the spectrum data, we take the Fourier transform, we found our pressure and displacement sensor peaks. And on top of everything, there's yet another innovative part here, which is the dynamic mechanical analysis, which hasn't been implemented with micropipette aspiration technique, that allows you to measure the frequency-dependent properties of the samples you're looking at. So we oscillate the pressure sensor and the displacement, and we look at the phase lag between them, and with using a simple formula, which is giving you the storage and the loss moduli, you can extract this information over a certain frequency range. So we couldn't go beyond a certain frequency because of the limitations in the pressure sensor side, so the microstring side. But still, what we observed is quite nice. It's unique itself. And one more thing, but you can do, usually what you do is to get the Young's modulus is you get the displacement and then the force, and you find the slope. This is your Young's modulus. But here you can also see the same thing without doing too much of effort, assuming that the storage modulus at a very low frequency range can be also considered as an elastic modulus. It can be comparable or similar. And we have confirmed it with the literature indeed by looking at the data that we got. And also some numbers to show you that we are also capable of finding the quantitatively elastic modulus numbers of different samples. And by looking at the literature, we also confirm that these numbers are well within the range that people have measured so far using different techniques. With that, I would like to thank you all. And especially, as I said, this guy is the guy behind this whole work. He's a very talented person. And also Optics 11. They are the major driving force, and very good news is that they are planning to turn this idea into a commercial product if you're interested, so you can always reach them. And I would like to thank you all for your attention. If you have any questions, please. I will, yeah.
Thank you, Imran. We are running out of time, but it's okay because we have this room after the webinar where we can discuss everything. So thanks a lot for your presentation, and I will see you later in the other room. Yeah. And now I would like to introduce our next speaker. That is Dr. Kanupriya, and she is an application specialist at Optics 11 Live. She has a double PhD in biomedical engineering at the Polytechnic University of Catalonia and also the University of Sarundas. And she's been doing, for a long time, she's been doing research in the bio-nanotechnology field. And now she's going to talk about the nano-indenter technology from Optics 11 Live. So thanks a lot for being here, and you can go ahead and share your screen.
Thank you. Thank you so much for your kind words. And thank you so much, I would say, Dexter and Imran. That was a lovely introduction. I learned quite a lot. Like they say, learning never stops. Today, I just want to give you my 10 cents as a representative of Optics 11 Live. Mechanics matter, and how can we help you? So you can know me as Priya. That would be easier a bit. And I was going through this editorial of communications biology, and where Marco wrote, "Mechanobiology is the elephant in the room. It has long been overlooked, despite the fact that biomechanics is known to influence many functional features of biological life." Funny, isn't it? Because in our everyday to-day life, we even buy the fruits by actually doing mechanobiology, seeing if avocado or banana or apple are ripe enough to be ready to eat, or it's good to keep it for 1 week. Literally nano-indentation in the day-to-day life. Now, thinking about mechanobiology, when I came from biomedical and cell culture field of scaffolds, porous scaffolds, and so much more, the first question I asked myself that how the root question would be about cell mechanobiology would be like how cell actually differentiates, matures, spreads, and so much more. And everything is influenced by nothing but the mechanical properties and forces of the surroundings, the shear forces, the blood flow, and so much more. It not only serves its physiology that the stem cell is going to turn into which particular cell, but also it actually initiates that how the organization of the whole structure would be, how the orchestration would be in the space and the time. Now, is it just cell? I don't think so. It's the extracellular matrix around it, which is providing the environment to the cell on its own, right? So coming to this 2D and 3D mechanobiology, these scaffolds, this extracellular matrix are nothing but like hydrogels that are trying to mimic this. The scientists in the in vitro life are really trying to come as close as they can to the native tissue so that it could be implemented in an in vivo and with a great success to the clinical trials. Now, this goes from the field of wound healing, skin tissues, you can call anything like regeneration, engineered tissue, drug delivery, drug encapsulation, cell therapy, and whatnot. Like list goes on and on. Now, we are already doing a lot of mechanical testing at a nanoscale, at a bulk scale. But like Dexter already mentioned also, what is it that happens at the micro scale? If my cell is going to be sitting in a hydrogel, is that hydrogel on its own good candidate to nurture that cell? Because if the host, the hydrogel, the scaffold, a tissue is not well, then a cell is not even going to spread. Forget the growing next day. Now, to sum up with this, I have a few questions that I would like all of us to think about from wherever you are joining today. The question would start with, do you work with cells, hydrogels, extracellular matrix, tissues, organoids, spheroids, any sort of engineered tissue? Because even if I don't see your faces, I know many of you has answered yes for at least one candidate. Now, do you do mechanical testing? Well, probably yes, probably no. If no, then there is a missing piece. And I would like to offer you a solution here by sharing our 10 cents of knowledge that how you can find your missing piece, how you can make your research, take it to next level, literally by finding the answers that you require to make sure that your cells are alive next day, your 3D scaffolds are degenerating at the right time for the drug release, or they are supporting the whole structure and the network around it and behaving like a native extracellular matrix on its own. Now, I don't need to dive into optics level life technology because there has been a lot of introduction on that. But just to start with a brief intro, like what is this technology? This technology is very basic. We use optical fibers. There is a probe that comes in contact with the sample at the bottom. There is a cantilever bending, and based on this bending, we are detecting how much deformation there was. Now, what is this deformation? This deformation is just to understand how stiff my sample is, what kind of load it can handle, and what kind of environment it would provide for my for whatever purpose you are doing research for, my cells, my hydrogels, my tissues, and so on. Now, what we can do, to sum it up, just a perfect introduction with the first presentation already, there is a displacement control. We can control the load. We can control the depth because biomaterials are depth dependent. You are not going to find the same stiffness at nuclei in comparison to cytoskeleton, right? Now, this data can be seen in the terms of static profile or the dynamic profile, which would lead to the information in the terms of the viscous elastic parameters. Did you know already? Maybe if not, because I would like to share it. Two materials with the same stiffness does not mean that they have same viscous and elastic modulus, loss and storage modulus, right? Now, what we can provide. We have probes from a softer stiffness from like 0.0 to 5 and then to 300 Newton per meter with a variation of the tip radius and the spherical tips. But the main important thing above all the information that it is that our nanoindenters are here to cover a wide range of 10 Pascal up to 1 gigapascal, from nanonewton to 3 millinewton forces. That's all that is required, because that pretty much covers a lot of soft biomaterials that are used as a ECM, as a scaffold, as a base, as a hydrogel, or even the drug encapsulation, polymer gels, and so on. Now, what are these nanoindenters? To begin with, I'm only going to focus on one side, but to give you introduction, there is a benchtop Puma, which is perfect for, inspired by a coffee machine actually, but it is perfect for the samples that are like, right, let's say maximum we can go up to 50 micrometer. And then we have Chiaro that can be compatible with any microscope. It could be any of your in-house microscopes, high-end microscopes, which if you prefer that. But then we have a new addition that is Pavone. Now, what is this Pavone? Pavone is a high throughput mechanical screening. It comes with high force precision. With a combination of 2 well plates, you can select whichever you would like, 6 with a 96 or 384. We have temperature module and so much more. Now, why there is this need of high throughput, or why this is like a next step in the mechanical testing field? Now, the beauty of the high throughput is that usually when we work with cells, we are actually always working in a lot of well plates, even 24, 96, and so on. And to save your time and to save the, if it's live cells and so much more, we would like to have this high throughput technology that can put a whole well plate in one go. Could you imagine that? Because now it's possible. This is one of the sample experiments that we created. We combined a 96 well plate with a 6 well plate. We had a series of functions that we designed as a part of the long experiment. We have automatic move, auto-find surface features. We have matrix scan done in each well with dynamic testing as well. Now, this is possible. We can run overnight experiments if required, and whatnot. Probably in long run, week-long experiments, that's definitely not far from where we are right now. Now, is this it? Because not. Once the experiment is done, what we are expecting quite a lot of data, right? So we can do, if we did the matrix scans, we can actually map. So this is one of the samples we have tested of a swelling hydrogel, which is swelling over the time, and you can measure its Young's modulus changing during the swelling phase of the 30 minutes or 40 minutes. And then there is a topography, how it is changing. So the auto-find surface function is adapting with the change over the swelling of the sample itself. Not only this, we are already measuring like organoids, hydrogels, tissues. You can do single cell monolayers. But one very beautiful part of the whole setup is that, which I personally love being a biologist, the ease of it. So what is that ease? If you look at this picture on the right, you have many multiple cells. You can literally just press which cells you want to indent, which you would like to indent, and the software will save the coordinates and it will run for you. Now, this is not just limited to the cells. Imagine you have a, let's say a tissue where you want to do a random testing at the random spots. You can literally select 5, 6 points. The system will save the coordinates, you save the experiment, and after that, you can run the whole experiment and it will be done for you. So the topographies of the organoids, the spheroids, everything is doable. Everything is already functioning fully. People are already using it. And this is definitely something making that, finding that piece to the puzzle. It's definitely doing the job at the moment. Now, I'm always curious that, is this the possibilities of indentation and image? Is it just limited to where we are? That is our research labs. Because I think when we have a success at a smaller rate, then there will be a mass production. It could be hydrogels or the scaffolds, and so much more. And for that, this could be a next step for the quality control. So, for example, we got this commercialized hydrogel from Matrigen. It's 96 well plate with a gradient hydrogel going from like really 0.2 Pascal up to 50 kilopascal. We measured the whole thing with one single probe. And the beauty is that among the replicates, there is a beautiful like reproducibility among each value. You can see how with such ease, the whole experiment was done with one equipment. Now, maybe you have got excited, or maybe I have not been able to communicate how interesting and how impactful this can be for faster, quicker, and a successful research. But just to give you a short sneak peek, how it really looks like, how it really works, here is Pavone in the action. So you look at the Pavone, I open the lid, you can see there is two combination of well plates. Let's say I want to move to A2 well, move to well, I press save, okay. And the stages will move to the respective well with the smooth and the ease that is required. Now, this is the same way the whole experiment would be designed. You can go from one well to another. You can do matrix scan. You can do dynamic testing. You can do indent control, load control, whichever control would be of interest to you. Because in the end, mechanics matter. Time is changing. And with 1 decade already gone from so much we could achieve already, I think the next decade is going to be like astonishing in the terms of research. So with that, I would like to say thank you very much for listening to me. And I hope I could actually add something to what you already know.
Thank you, Priya. This has been a really nice presentation, and definitely tools that are going to help a lot on the current research, because we see more and more high throughput is a solution that many labs are using now, and it's advancing very, very fast. So thank you. We will see you later in the room for questions. Sure. And now I will move to our next speaker. That is the last one for today, and it's Jeroen Kolle. He's the product application specialist in Confocal NL, and he has a background in biology and nanotechnology, and has several years of experience in microscopy. So he will be talking about the benefits of the real-scan confocal microscope for mechanobiology studies. Thank you, Jeroen.
Thank you, Noelia, for the introduction. So, looking at the time, I've condensed my presentation a little bit, but I kept the beautiful images in, so that's what we are here for, I think. So my name is Jeroen. I'm product application specialist at Confocal NL. Today I will be talking about our new product, the RCM2. And as a tool for mechanobiology, we saw Dexter's presentation using the RCM1 with beautiful complementary technologies that I think will really strengthen this field. Okay, so what do we do, and what is the RCM2? So at Confocal NL, we make the rescan confocal microscope, which is here, the orange box, which attaches to the C-mount of any research grade microscope. And what you can do with it is actually long-term time-lapse imaging of living cells in very high resolution without causing phototoxicity or bleaching. On top of that, it's also very easy to use and straightforward. Okay, so the RCM can be coupled to a microscope as an upgrade, but we also offer complete systems, including the microscope, table, computer, everything included. So the specifications of the RCM, it's a camera-based point scanning confocal that achieves 170 nanometers in the raw image and can achieve 120 nanometers after deconvolution. Z resolution of the microscope is 500 nanometers and 350 nanometers after deconvolution. Then the quantum efficiency, this is the detection efficiency of the system, is very high, up to 95%. And this is actually more than twice as high as the modern standard in confocal microscopy. we can scan at about 2 frames per second at 512 by 512 pixels. And we use 4 lasers for excitation. So we use 405, 488, 561, and 640 as standard wavelengths for excitation. And to get this 120 nanometers, you can use a range of objectives ranging from 40 to 100x 1.4 NA. So these are oil objectives, which achieve this super resolution. And what is special is actually this number here, 40x. So we can do super resolution imaging over a very large field of view. So here are some nice movies that I was talking about. This is a HeLa cell stained for actin filaments using the LiveAct probe by EBD. So you can really nicely see all kinds of dynamics going on here. You can see these filopodia moving around. You can see the cells sensing in the environment. And this all without causing any bleaching or phototoxicity. As I mentioned, RCM can do super resolution imaging over a very big field of view using a 40x magnification. So here you see a movie of dividing UVEX, which are stained for actin, tubulin, and DNA. And you can see here the field of view that you can get on a 40x magnification. So all this resolution throughout the image, here we zoom in digitally, all this resolution throughout the image is 120 nanometers after deconvolution. Right, moving on. You can take this to an extreme level. So this is actually one of my favorite data sets that shows cells moving around. The mitochondria are stained, and we image these cells for 61 hours. So 61 hours of continuous imaging with 1 frame every 10 seconds. So in total, we acquired over 22,000 frames. And you can see these cells are completely happy. They're moving around. This one now goes in mitosis. And this is something that is really hard to do in conventional confocal microscopy or even microscopy at all, because mitochondria are very sensitive organelles that really don't like any light. But in RCM, as you can see, these are happy. Okay. So one more image on the large field of view. So here we see a 40x image, 5,000 by 5,000 pixels, and this is digital zoom. So you can see that these chromosomes, these spermatocytes are actually resolved. So you can see like these individual bands, and this is not visible in confocal microscopy, in regular confocal microscopy. So you can really nicely observe them here. And of course, you can do deconvolution and take this even one step further. So here you see deconvolution performed with microvolution, and you can really nicely observe these the spacing between the chromosomes. Of course, confocal microscopy is really all about 3D. So here we see a HeLa cell stained for actin in green, tubulin in red, and DNA in blue. And you can nicely move around the cell. It was acquired with a 100x oil objective. So this is just looking back at the question that is what was asked to Dexter before. RCM is really suitable for deep 3D imaging. So here we see another example of a mouse blastocyst being imaged with a silicon objective lens, and there we were able to go all the way through the 130 micron diameter ball sphere, if you like. So this is this shows high resolution imaging deep inside the specimen, 130 microns in, and this objective allows you to go even deeper. Okay, this brings me almost to the end of the presentation. So just to summarize, RCM2 is a super resolution technique. It can achieve 120 nanometers in the deconvolved image. It's very sensitive because of the camera. So we already demonstrated data sets acquired with only 50 nanowatt of excitation light. So this really sets a new limit on what is possible with confocal microscopy. The RCM is very robust, easy to use. We are flexible in terms of software, and versatile in terms of applications. So we can convert your existing microscope with RCM technology, or we can supply you with an entire system. This is the end of my presentation. Thanks for listening, and I'm happy to take any questions in the breakout room later.
Okay, thanks a lot, Jeroen. I think we had a very nice webinar. In general, seeing how microscopy and imaging technologies can complement the studies in mechanobiology and to get more precise results and like answer all the research questions that we may have. And now I would like to thank you and to explain about our tool that we have a second room now. So you will see the offer to join and to click in this link, and then you will move to a different room where you will see the name of the speakers, and you can move yourself around with using your mouse and join different conversations. So you can visit each of the speakers, or you can either go to the tea room for social conversations. And it's very easy to use. You will get familiar with it very quickly. We will have also questions for ice breaking, and I would like to see you all there. And I would also like to encourage you to follow us on our social media, because it's where we are posting all our webinars, and also visit our website for more information about the products. And also, if you see the replay of this webinar, it will be there. So thanks a lot for joining us today, and I will see you now in this after party room. I hope that you can join us. Thank you.
See you. Bye-bye.